All right, hello everybody, and welcome to Engineering Showcase, Fighting a Global Pandemic, the Frontlines of Innovation hosted by the UMass College of Engineering in collaboration with the UMass Amherst Alumni Association as part of UMass Amherst Homecoming 2020. We are so excited to have you all join us today.
Hi, everybody. I just want to start by introducing the team behind the scenes today. So my name is Karen Schultz-Battistoni. I'm the Director of Alumni Relations in the UMass Amherst Alumni Office. With us is Alexandra Lazaro, an engineering student, class of 2023, and M.K. Young, Assistant Dean for Development from the College of Engineering. And we'll be introducing our panelists in just a moment as well. Just to give you a few details about today's session, if you want to connect with us, you can chat in the lower box. So there's a chat box there. Feel free to put any questions, any comments in there. You can chat either with the whole audience and the panelists, or you can just chat with the panelists. Either way, we'll get the question. And then Alex will be taking those questions and bringing them forward to the dean in order to get them asked of the panelists. So just to be aware, for tonight's session, we are recording this session to make it available for viewing at a later date. By participating in this session, you are giving consent to being part of a recorded session. All attendees, you will have your cameras off and your microphones are also muted. So you don't need to worry about any of that. All you need to do is chat with us in the box in order to communicate with us. So without further ado, I'm going to turn it over to Alex to introduce our moderator.
Thank you, Karen. I'm so glad to be here and be part of this homecoming event with the College of Engineering. As Karen mentioned, I am a biomedical engineering student at UMass, and it's so great to be here with many alumni to join us for this conversation with our faculty, alumni, and industry panel. As Karen mentioned, please enter questions you have for the group into the chat box at any time during this session. And without further ado, I would like to introduce our moderator, Dean of Engineering and faculty member, Sanjay Raman. Dean Raman joins us from Virginia Tech, where he was Associate Vice President for the Virginia Tech National Capital Region, President and CEO of the Virginia Tech Applied Research Corporation, and a tenured full professor in their Bradley Department of Electrical Computer Engineering. From 2007 to 2013, Raman was a program manager in the Defense Advanced Research Projects Agency Microsystems Technology Office. Raman earned his doctorate in electrical engineering from the University of Michigan, Ann Arbor in 1998 and joined the electrical and computer engineering faculty at Virginia Tech. Prior to his doctoral studies at the University of Michigan, Raman served as a nuclear-trained submarine officer in the US Navy from 1987 to 1992. He's an elected fellow of the Institute for Electrical and Electronics Engineers for leadership in adaptive microwave and millimeter wave integrated circuits.
Thanks, Alex. I really appreciate the introduction and all the kind words. It's really great to be here with you and the rest of the team. I've got to give a shout out to you as one of our new biomedical engineering students. You've obviously picked an important discipline within engineering that, as I think, yes, all of us will learn during the course of this discussion, the intersection of engineering and medicine and healthcare is really what we're here to discuss. So today I'm hoping that we can have a very interactive and robust discussion about some of the work that's going on here at the University of Massachusetts Amherst, and along with and in partnership with our friends in industry, as well as many of our alumni. And so we are going to talk about a couple topics today. The first, I'll just mention a couple of early response actions that happened in the College of Engineering in collaboration with our colleagues in the healthcare disciplines. And then we'll talk about some new research that is building upon a number of innovations that have happened in the College of Engineering and how they may help contribute to our global response to the pandemic and certainly future pandemics as well. And we'll talk also about the front lines of healthcare and front lines of medicine and the intersection of those front lines with the innovation process. And so I think you'll hopefully really enjoy and be able to participate at length with the panelists, and we'll have a really interesting set of discussions. So let me take a couple minutes and talk about some things that happened in the very, very early days of the pandemic in mid-March. I think many of you, we just gone into lockdown. Many of you were in the same boat, just kind of caught by surprise by, the rapidity of the spread of the pandemic and all the potential negative consequences associated with that, both in the healthcare space as well as in the economic space. We're all just dealing with this, many of our faculty and our students and our alumni were thinking about what they could do, and there were a number of interactions that we had with our colleagues in medicine and in nursing and in public health. And some of those discussions led to some very interesting projects that occurred within days, really, of the shutdown. So on the left here, you see Shira Epstein, who's a lecturer in the College of Engineering and Electrical and Computer Engineering in particular, and also director of our campus makerspaces. And the Electrical and Computer Engineering department had been contacted very early on in the crisis by nurses at Bay State Hospital. And they were finding that, as you recall, there was tremendous lack of PPE at that time of the crisis. And they were finding that they were having to go into patient rooms to take readings and to operate ventilator systems and other systems within the hospital rooms of the patients. And each time they were having to gown up in PPE, and then they were having to discard it, and it was resulting in a lot of waste in the PPE. So a very simple question they asked our engineers: can we just lengthen the ventilator cable so that the machine can be placed outside the patient room, and we don't have to use up all our PPE just going in and out and monitoring the equipment? The answer was yes. A very, very rapid response was done on basically, somewhat simple from a technological point of view, but just, how do you lengthen this cable? Is there sufficient signal integrity between the ventilator unit and the controllers? Some interactions were done with some industry partners in the area, including one of our alums from the ECE department, to figure out how manufacturable these cables could be. We brought on board an industry partner to help manufacture this, and within days, we had a number of ventilators up and running in Bay State that were able to be operated remotely and made a much easier and more efficient process for the nurses at the hospital to be able to, interact with their patients and the patient monitoring systems. At the same time, there was a tremendous shortage of face masks and N95 masks and face shields and PPE in general. And a couple of our faculty members here on the right in the Mechanical Industrial Engineering department started interacting with nurses. And they found out that there really was a need for very low complexity, very easily adjustable, no fog, disposable, low-cost face shields that could be deployed very rapidly. And if you recall at that time, there was a lot of work going on can we use 3D printers? Can we use additive manufacturing? And it just turned out that doing 3D printing, even for relatively simple structures, just couldn't be done at scale at that time, particularly with the capabilities that we had on campus. So these faculty members, Megan Huber and Frank Supp in Mechanical and Industrial Engineering, came up with really a very, very simple design that just relied on flat sheets of no fog polymers. And they could be, the pattern could be cut out using either a laser cutter or a die cutter. And the mechanical design was done in a way that it could just be simply wrapped around the person's head, and it fit. It could be adjusted for fit. An N95 mask could fit under it very nicely. Very manufacturable, very rapidly. And within days of the crisis, thousands of these were being manufactured at some scale. A patent was filed, and we have opportunities to potentially turn this into a product in the future. So these are some things that that happened very early on. There are other examples, but what I really want to point out is that those innovations really occurred because there were very robust discussions between healthcare providers, frontline medical workers, and engineers and scientists. And so we learned really firsthand what the problems were, what would be the solutions that could make a difference in those situations, but also some of the human factors things, what was going to be comfortable, what was going to be usable in that environment. And all these things were done really in days because we had some very, very open and helpful discussions with the frontline healthcare workers. And so I think that's going to be a theme throughout this discussion, that we we work together in partnership with the frontline healthcare workers and medical professionals in order to get to a solution that would make a difference. So let me very quickly here summarize a couple of the things that we're going to talk about more specifically over the course of this afternoon. First, we'll be hearing from Professor Jonathan Rothstein, also in mechanical industrial engineering. He has developed, using some basic fundamental research that he's been doing in the fluid dynamics area, a new quote unquote breathalyzer approach to creating a testing platform for coronavirus. And you're going to hear about some work that he has funded by the National Science Foundation under the Rapid Response Program, and he'll be talking with us about that in a couple minutes. We'll also be hearing from Jessica Schiffman, who's an associate professor in chemical engineering, who has also received some very recent rapid reaction funding from the National Science Foundation to look at how we can engineer filtering masks differently. And in this case, it's a Bioinspired technology that is based on the pitcher plant and provides a very efficient means for capturing airborne droplets of corona that may contain the coronavirus. And lastly, we're not going to hear specifically about this project, but we did want to let you know another effort that's going on that's currently being considered by the National Institutes of Health. And this is looking at wastewater. We have a very strong wastewater program in the Civil and Environmental Engineering Department in the College of Engineering. And this is using wastewater effluent streams to be able to identify where there might be an outbreak happening before the testing regimen actually catches up with that. So you can see in this graph, by doing measurements on different signaling pathways in the wastewater stream, you can detect potentially a potential outbreak of coronavirus before everyone in that particular location can possibly be tested. So we're not going to hear about that in detail in this afternoon's discussion, but again, another innovation that's happening in the College of Engineering that could lead to potential solutions to combat this or future pandemics. So next slide, please. So it's my really, really great pleasure to introduce our distinguished panel this afternoon. I've already mentioned a couple of these folks. So Jonathan Rothstein, professor of mechanical industrial engineering, expert in fluid dynamics. Jessica Schiffman, who is an associate professor in chemical engineering, expert in biomaterials. And then two of our close colleagues, Bob Steininger. He's a senior VP of chemistry, manufacturing controls at Surface Oncology, and he's going to bring a perspective of, first of all, from the industry perspective, how can these technologies that are being developed in university laboratories make it into products? All the issues associated with developing an innovative product, getting it evaluated by the FDA, turning it into something that can actually be used for patient health or for medical use. And finally, Anne-Marie Tesramata is actually one of our alums, a 1977 grad, also from chemical engineering, but then took a turn and went into the medical professions, MD, emergency medicine specialist. And she's going to talk about how her experiences on the front line as a healthcare worker, how engineering led her to be able to be successful in that field, and also join the conversation about how frontline healthcare workers and medical professionals can be part of the innovation process and lead to these kinds of technologies. So with that, let me stop talking there and turn it over to my colleague, Jonathan Rothstein, who's going to talk a little bit about himself and his technology.
Thank you, Dean Robin. Thanks for the invitation to be on the panel. And I'm looking forward to a really fascinating conversation and discussion this afternoon with everybody out there in the audience. My name is Jonathan Rothstein. I'm a professor in mechanical and industrial engineering. And I've been in the MIE department now for almost 20 years. So some of you who are alums out there, reasonably recent alums may have actually had me in class or remember me from way back when. So my expertise is in fluid dynamics. That's where my research is based. And so what I'm going to do is I'm going to just give you a sort of a brief introduction into sort of the technology that we've been developing and sort of talk about the sort of the evolution of how we started and where we are right now. So what I have here is that sort of the story starts probably about in March or May, between March and May, when I sat down and was talking with a colleague or a friend of mine. We were all graduate students at MIT together, and we were just talking to each other and feeling a little bit like we wanted to make a difference and try to move in that direction. So what we came upon was working with a technology that allows us to collect exhaled breath and use condensation from exhaled breath to collect and then eventually diagnose COVID-19 that's actually in your breath as you breathe out. So one of the major sources of COVID-19 is the small particulates that end up in the air. That's why masks are actually really quite effective at slowing the spreads, because they reduce the plume of virus that's emitted as you breathe outward. And so our idea here was to attempt to actually capture the virus directly from your breath. And that will allow us not only to measure whether or not you are infected with COVID-19, but also to determine the degree of your contagiousness. So we can not only count and determine whether you are infected with virus, but how infectious you are to the community around you. So we started this process in May. We started first by going after small grants. So we went after an NSF grant, which we were able to receive part of the RAPIDS program, which is designed for, as it sounds, rapid funding of ideas. And there was a strong focus at that time to fund as much new science in COVID-19 research as possible to try to address the pandemic. And then we continued working and developing the technology. Went after and got another grant from the NIH for actually commercializing the system and the technology, and have spent the summer and now part of the early fall in developing a company, coming up with a finalized design for the collector, and for developing a way to take the collected liquid off of the surface of this plate. This is a cooled plate with a specially modified surface that allows you to collect the droplets effectively. Take that, send it out for RT-PCR, and also put an inline detector so that we can make a point of care system that can detect the virus in about 10 minutes using what's called FISH, which is fluorescence in situ hybridization technique. And so we are at this point, in the process of, we've incorporated the company. We have a couple patents being filed for this technology. And we're in the process of human trials, which will be starting in less than a month out in California, where my colleague is at UCLA. So that's sort of just a quick introduction to the technology that we're working on. I'd be happy to talk all sorts of details to the limit that I can with any questions that you guys have in the audience moving forward. So I guess
Okay.
I'll pass it back to you, Dean Rahman.
Okay. Yeah. Thanks, Jonathan. So I think we'll hold questions for the moment. We'll do the introductions, and then we'll have a sort of a joint discussion with everybody about these specific technologies as well as sort of the whole innovation process. So let me kick it over to Jessica Schiffman, Associate Professor of Chemical Engineering, to talk about the pitcher plant.
Great, thanks. I'm also so excited to be here to share what we've been doing. We've been making bioinspired membranes, but why didn't I come up with the name BlowFISH? It's so great. Anyway, so let's take a step back. So my lab we're in the chemical engineering department. I've been at UMass since 2011. We're actually a material science group. We make new materials, mainly out of polymers, for a wide variety of human health applications. For those of you that haven't been on campus lately, which also includes myself, we're in this beautiful new Institute for Applied Life Sciences. And below that is a picture of my wonderful research team. We have many different graduate students and undergrads working together, although right now we're only working in groups of 2, 6 feet apart. So we make polymer materials mainly for a wide variety of human health applications, from biomedical implants to improved contact lenses to paint, and then also membranes for water purification. So the story of how we started doing membranes for air purification, for capturing of COVID, really is a story about one fantastic undergrad, Jared, who's actually in this group picture in the back row with the long hair. So yeah, he had been working on these membranes for water purification and said, I'm really interested in improving air quality. So fortuitously, about a year and a half ago, 2 years ago, he started an independent honors research senior thesis project that was really looking at some of these nanofiber samples, or like very, very soft tissue paper, and how they themselves could be used to clean particles out of the air. When the pandemic hit, I think us engineers, like many other people, felt like, what can we do to help our community? As Dean Rahman said, many of us started with these quick reaction projects. So at first we had all the materials that we needed to help the hospital staff make the serums to react the COVID analysis solution. So at first we were actually making serums and shipping them to Boston to analyze, is this COVID positive or negative, this human samples. And then over time, we actually realized that this air purification membrane that this undergrad was working on could actually be adapted in order to help identify if COVID aerosolized particles were present. So we were already, making these materials to, say, collect solid particles, but how could they be adapted to collect liquid particles? And so here is where we really did look to nature. So I'm not a biologist. I actually haven't had biology since high school. But what the pitcher plant does that we know about is that it has a liquid layer, and the liquid layer is slippery, and it can help trap things and keep them where we want them to go. So this is really where our research started, and that we took these kind of tissue papers, coated them with this slippery liquid, which doesn't feel slippery to us. It's just a very thin layer. And this way we can have a mask or an insert into another air purification system. It's going to collect these aerosolized COVID particles. And then those particles, or the aerosolized virus, actually stay viable. So we can actually take those COVID particles and see, are they actually a viable virus, or is it a dead virus, or is it not a virus at all? So this is another way that we can take the mask that we're wearing and simply breathe into it, collect what's deposited on it, and know if we ourselves are carrying around, a virus that's positive for COVID. This research is also supported by the National Science Foundation, with some collaborators in Maine, by a very similar, starting grant to what Jonathan Ronstein had mentioned. We've already filed some patents on it, and we're really, just looking forward to seeing who else wants to collaborate with us on developing this technology further. I think before I hand over to the next person, I'll also say that during these really challenging times of COVID, both in our research and actually, in our classes, our students have been really working hard. And you might be wondering what you can do as alumni to pitch in. We've had really excellent talks from all sorts of alumni coming back to either give a guest lecture about what you're working on now or other ways to contribute and what people have done with their different engineering degrees. So even though these are really hard times where people could feel really disconnected, we also have really been able to take advantage of all these technologies in order to reconnect with people that we haven't talked to in a while. So I guess with that, I will hand it over and look forward to some of the questions later on. So thank you.
Yeah, thanks, Jessica. Hey, what your slide just prompted me to mention also that I think for the benefit of the folks who aren't on campus, we have a pretty substantial research set of research efforts that have been going on since probably May, June timeframe. It was a priority for us to restart research in a safe way, obviously, in a way that we could maintain social distancing and appropriate protections for the researchers in the laboratories. But it was very important, not just for the specific projects that are potentially solutions toward pandemic-related problems, but the whole research enterprise in general needed to keep going and needed to keep our graduate students and our faculty moving forward on their different projects with different sponsors. So there was a very significant effort in a number of committees at the university level to figure out how we could ramp research back up in the safest way possible, be compliant with the state guidelines that were coming down from the governor's office of the different phases of the pandemic. And so we were able to basically get up to upwards of 30 to 40% research activity in the very early months of the pandemic, which really was important to allow faculty members like Jonathan and Jessica and their graduate students and undergraduate research students to keep going on these kinds of efforts. And we're at the point now where we're in the neighborhood of 70-75% research activity back on campus. Again, following all the appropriate protocols. In many cases, laboratories are being operated in sort of a shift work mode. So a set of graduate students or postdocs will come in and work for 6 hours, and then another shift will come in and do their part. So that's been a pretty important accomplishment by the campus. And as we get through the pandemic and move on to the other side, we've been able to operate the research labs at a pretty high level, and that's been important for a whole range of research efforts, some of which you're going to hear here. So let me then turn it over to Bob Steininger, who's going to talk about the industry perspective of this and of course, his own experience and how he got here as well.
Hi, my name is Bob Steininger, as mentioned. And a little background on my own work in biotech. I've actually been in the business for almost 40 years and sort of grew up with biotech. As you think about what biotech was in 1980, there wasn't very much. And quite frankly, I actually grew with some of those companies, including one that I started with, which was Genetics Institute, which actually went from, 60 folks when I started to nearly, it got bought by Pfizer. So it's huge right now, but it's certainly in my time being there, actually expanded to be 2,500 folks. My own background really spent a number of different disciplines related to biotech. And it really goes from process development to manufacturing to regulatory affairs. And what that is probably the most important aspect, because any aspect, any device that is used in medicine has to be approved by regulatory authorities. And the two examples here are not necessarily implantable, so they're devices, but not implanted. It should be a little easier to get through the regulatory aspects related to how it is. And I think you'll just have to show, quite frankly, that they are reproducible and be made in a consistent fashion so that you can get that sold. And that's where the regulatory focus will be on such devices as we move forward. Obviously, one of the nice things of UMass certainly has a broad spectrum they can focus on, and certainly devices are part of that, particularly with the depth of engineering that UMass has. But certainly they have also expanded substantially in the other areas, and that nice building that was shown there certainly goes beyond just making devices. It certainly has the capability to actually look at making drugs or at characterizing drugs or figuring out what new drugs can be done. And certainly a lot of my background has been in that area. How do you take a good idea and make it into a medical instrument or drug that can be beneficial to people? And again, devices can be done relatively quickly as long as you can find the money to do that. And there I would say alumni from UMass might be very helpful in sort of linking up the folks that do have some money that might want to look at new ideas. And that's something that I think certainly alumni could be very helpful related to new ideas coming from UMass, but certainly other things certainly have been done and can be done related to drugs. And there's really how much can you help in actually taking drugs that are not well characterized and be able to do that. And I think UMass has the ability to do that as far as moving a drug from an early phase 1 candidate into a more larger trials of phase 3. And that's really the main issue, whether it's a device or a drug. The real focus of really most of Cambridge and Boston is getting that ideas from UMass and the other universities to actually result in aspects, whether it's device or drugs, that can be used directly in patients, locally and certainly internationally. So that is where I have been, and that has really been my focus through the 40 years.
Oh, thanks, Bob. I think one of the things that we really want to explore with you when we get back around to the Q&A is really how that ecosystem works and how professors like Jessica and Jonathan they've got this great idea, they patented it. Maybe they've even, they've gone the step now that Jonathan has to incorporate and start a company. What are the next things that happen? So I hope we'll be able to spend some time picking your brain about how that whole investment, regulation, industry partnerships, and so forth can turn into real products. So let me finally introduce you all to my great pleasure, Anne-Marie Tesramata. Again, as I mentioned, 1977 grad in chemical engineering from UMass, and then went on, got her MD, and is an experienced emergency medicine medical physician. And she's going to talk a little bit about how she got there, and then we'll spend some time hopefully also picking her brain about how frontline healthcare workers and medical professionals can influence the innovation processes that we're talking about here. So, Anne-Marie.
Thank you, Sanjay. And thank you for inviting me. I'm honored to be here and part of this showcase. My journey to engineering started with actually interest in biology in 10th grade. And yet I was also interested in engineering, was a daughter of an engineer, and landed in chemical engineering, both as a way to learn engineering as well as to learn chemistry and all those other things you needed to get into medical school. I worked with Dr. Stanley Middleman in my senior year on membrane research, and we were working with urea and developing membranes for dialysis, and went on to work with Sidney Loeb that summer, who was the father of partially reversible osmosis, and we were working on generating energy waterfall with chemicals and membrane systems. But from there, I went to medical school, and it was a demanding life, and ended up training in internal medicine and becoming interested in emergency medicine. and spending my career in emergency medicine. I think it was a natural kind of transition for me because engineering leaves you with a real sort of problem solving mindset, a very deductive reasoning, a lot of critical thinking. You're always looking at data, you're always making decisions. And that type of thinking that I kind of was trained in engineering kind of translated well to my medical career and my medical career in emergency medicine. So it's been sort of a great transition and a great platform for me to work from. I've worked for over 30 years in emergency medicine and lived through many epidemics, illnesses, and coming over the horizon, from the early one of Legionnaires to AIDS to MERS to the H1N1 pandemic to Ebola, and now to SARS-2 or COVID-19. We've been presented with all the challenges that everyone else is presented with, but being essential workers, you know, our job has really been to be there 24/7 to keep the ER open. I think you'll find in medicine, particularly in a lot of critical care specialties and emergency medicine, you know, we're just taught to think on our feet. So we're coming up with sort of just immediate solutions and innovations as we work that day to conserve PPE, to do things differently, to figure out how many times you need to be in a room, how to communicate to a family, how to communicate to a patient, how to communicate to your team, and now have this whole new issue of safety with every communication on board. So you're dealing with a great team. You're wearing many hats as the leader, as the clinician, as the protector of your team, and you're looking for ideas everywhere. Sometimes the ideas come from the most non-clinical person on the team. It could be the housekeeper, the person doing registration. You really have to listen to everybody, and there's places for applications and technology everywhere. I think there's one question that, you know, I really like to hear I've really been thrilled to hear what you are doing. It has so many applications. And I think a lot of times you'll just have people saying, you know, if we could do this, if we could just do this, this is what we'd really like to do. We'd like to not go in that room so many times. So how can we not do that? You know, how can we conserve the PPE? I have to say, the first day that I landed back in the ER to treat a patient, the nurse said, okay, you need to go in and see this patient, and here's a disposable stethoscope, and here's a gown, and here's gloves, and here's a mask. And I came out and I said, okay, so am I supposed to throw this away now? This is not going to last. This is only the first patient the first day. So everyone on every line, from healthcare workers to EMS workers to grocery store workers, has an experience that can shed a light on areas where technology can be applied. And that's really, I think, where the excitement can be. I thought about Jonathan's rapid testing system, and I thought, wow, that's really great. And what that reminds me of as a clinician is a reverse inhaler or a peak flow meter. It's the same kind of physiologic action. You're just condensing the breath. It brings up a lot of questions. You know, how much virus can you get? How accurate it is? Wouldn't it be great if it was point of care, because then an EMS person on the street could use it and see a color change and identify a potentially risky person before they even got to the hospital. And that would create a whole new system. So there's a lot of excitement, and it's great to see everyone from A to Z working to invent new things. Our inventions weren't quite as elegant, on the streets, but people put their heads to the problem and created a lot of mechanical solutions, including intubation boxes, intubation tents, just simple things. Like you said, a simple mask can be, a simple plexiglass mask can be the easiest, best thing that you can have. So it's really, and it's a hard time, but it's an exciting time. I'm excited to be part of this program and talk about these things. I'm also very excited to hear about the membrane technology, and I think that I could imagine a lot of applications for that.
Thanks, Ann. That was great. So I just want to maybe ask one question here to you as we transition into the Q&A part of the discussion. So you had these experiences on the front line in the ER, or even just going back through your career. Were there channels for you to sort of communicate ideas out to sort of the innovation ecosystem? Was there kind of like a process for that, or did these things kind of die in the ER when you said, hey, it would be great if we had this, but never really went anywhere? Was there ways of making that happen from your perspective, or what could we do better, as innovators?
Well, I think people who were ambitious with an idea really took it places. And I think, a lot of life is sitting around talking and imagining and saying, oh, wouldn't it be great if we could do this? And it's true. Where does the idea go from there? One of the best inventions that I ever saw, which was very simple, is something called a splash shield. And a splash shield is just a plexiglass little cup that goes on the end of a syringe that we use to irrigate. And when that came out, we were just astounded at how great it was. And the guy that invented this would not be working nights anymore. So it's just amazing how a simple thing can be so great. I have to say, as a user, the plexiglass face shields are one of the easiest things to use. And in combination with N95, I felt pretty safe. I could put it on, I could take it off, I could wear my glasses. I think really the key is to know what the user is experiencing. One of the first things that people experienced, which was more the nurses than the doctors, I think, because they wore their masks longer, was their ears were hurting. The string was hurting their ears, and yet you needed to wear the mask. And they invented something to tie up those strings and really still maintain their safety. So everybody's mind is at work all the time. And we don't all have that great technological knowledge that you have that's patentable and invents things from materials. But everybody has a thought, and that can grow in all directions. So it's really interesting.
Yeah, thank you. I mean, I think that the interaction between the medical professionals and the engineers and scientists is crucial. I think if we can somehow create that ecosystem in a more sort of systematic way, we might really be able to do some cool things, maybe faster. I mean, maybe they would have happened anyway, but maybe we can get them to market faster.
Well, I think that's a great thought. And I think, I think there's lots of opportunity between the medical school and the multiple campuses of UMass now. And then the number of alumni that there are, let alone from the medical school, which was relatively new when I graduated from it. So I think and now you have a Department of Biomedical Engineering. And what I really didn't say was I really wanted to be a biomedical engineer when I started out, and you didn't have it. So it was a graduate degree. And I'm like, well, okay, I guess I'll settle for chemical.
Hey, it's never too late to come back.
Hey, you never know. I might apply.
Well, hey, let's delve into some of the questions here that have been submitted. There's a ton of questions actually, so I'll try to merge some things into a set of conversations. We may, so I'm going to apologize in advance. We may not hit every single question sort of verbatim, but there's a question from Sue Morgenstern here about tech transfer, investment, the university ecosystem for new technologies. So let me ask Jonathan and Jessica and also Bob to discuss kind of the process. You know, had this idea, Jonathan, you touched on this a little bit already, had this idea, patented it, got into some discussions to form a company with your colleague. What's next? How does that all work? You know, how does it go from the university laboratory into Bob's hands?
Yeah, so it's a relatively straightforward process. I mean, we have a great office of tech transfer, and they work with you sort of every step of the way, from the point of filing the initial invention disclosure to the process of going through the patent application. For us, at the point when we knew that we had a company, we have two provisional patents on our technology. So we were trying to figure out how to best license that. And so we were able to contact the the tech transfer office and essentially enter into a letter of agreement to get a license for a exclusive license for the patents that we had developed. And that's actually pretty, in general, if you're the inventor from the university and you go back to the university and say, I'm starting a startup company, we'd like to get a license for our own patents, they work with you really quickly. They're very efficient at it. They want to see you essentially develop your work and bring it to market because they know, of course, you're the one who knows it the best, and it will have the highest likelihood of making it, turning it into something that's profitable. So Robert or Jessica, do you want to follow up on that?
Jessica go first, and then I can say, well, next steps, because there are some next steps you're going to have to do in order to capitalize in your inventions directly.
Yeah, so from my perspective, I'd say I've seen, what Jonathan kind of has reported. Our tech transfer office is awesome so far in terms of my needs. I kind of wanted to just take a step back and talk about some really awesome initiatives that have been happening. So this last week was the Innovation Challenge run by the Eisenberg School of Business. And this semester, since I couldn't have undergrads working in my lab, I actually was approached by 4 undergraduate students who heard a talk given by one of my graduate students and said, this is really cool. We'd like to pursue this as an independent study and enter the Innovation Challenge. So despite being 4 undergrads in STEM, this 4 women team really, they came in second. They got the Viewer's Choice Awards. I'm sure there's many wonderful, people in business, but I was super proud of our STEM scientists to say, okay, we can go learn how to, sales pitch this. And then, okay, so what's happening next for them after winning this minute pitch in front of, serious investors and people that are really geared in for their success? They're actually entering this I-Corps competition, or I guess training program. So UMass is home to this I-Corps program, which will really train them in ways that probably, I never had a course on how to develop, the patent to something that could actually make money, because of course, how we do things in our lab, how the basic science works, how we make things on a one-to-one basis is not how things get scaled up. Beautiful engineering problem. So yeah, so there's many different ways, I'd say, both for helping the faculty learn how to do these things better, and then also even from the undergraduate level and graduate students as well. Yeah, so maybe Bob, you can tell us about what we have to look forward to now.
Well, I think one of the aspects is if you have a design that you feel comfortable, then how do you make it multiple times? And then who's going to pay for that? And really that comes back down to, are you going to go or do this yourself? And if you're going to do it yourself, to whom do you actually go and get that money? You know, and there's certainly VCs, and I'm just looking at it. I think there are almost 1,000 companies doing biotech in the Boston, Cambridge area. Of that, you know, there are certainly a large percentage of those are making devices that are used for biotech. And so there are VCs that are focused on that. And that's one way to do it. If you can then convince the venture capitalists that is something that has a reasonable plan. And this is where somebody within your company needs to put together that business plan and be able to sell that. You know, it's the typical elevator speech, which you're going to have to go to that someone to say, this is what you want to do, and this is going to make. And here it is. I think a good specific initial product, would you say this is what can be done? That's great. Then the question, well, how can that be done least expensively so that could be sold? And it could be, small company or a large company, but finding those companies that you could actually do, or the money really comes back down to really doing a little bit of search and finding the interest. And that becomes as much a sales job as anything else, because that's reaching out to people that have done it before or have money that would feel that this particular invention. And, I've seen that from VCs to physicians, right? That can actually be that. Some physicians had that capability directly, and they have connections to the folks that truly believe, okay, this is something to be done, right? But it's those connections are extremely helpful. And I don't think the regulatory is the issue. It's how do you make it inexpensive enough so it becomes valuable for the medical community and somebody can see how they can make money on it, right? And that's where it comes down to brass tacks. So it's those connections, I think, probably most important for you.
So Jessica mentioned
development person.
Jessica mentioned the I-Corps program, which we're an I-Corps site. We actually work very closely with MIT, which is a regional hub in I-Corps. But one of the key facets of the I-Corps program, lean launch type of mindset is the customer discovery piece. Like, how do you know there's a market for the thing that you are, it's great. We sit in our university lab and say, hey, we think this is awesome, and I'm going to sell 10 million of these things. But really, and this is maybe here again, this interplay between, the nurses and the physicians and the frontline healthcare workers and the products, right? Or the idea. So how does customer discovery, what do you look for as a VC? How well understood does the market need to be before you invest? And maybe the flip side question is how to get the healthcare professionals involved in that process.
At least my experience, it really helps to have a committed member of the medical community to say, this is how it can be done. And they tend to be very, I would say, vocal and also compelling to the other physicians within the group, right? So I think that's an important aspect. And then it's actually showing why it's that much better than what you already have, right? And, really a VC person can put together the effectively analysis to say, yeah, this is how we could do it, and this is the cost that needs to be made in order to do this reasonably, and maybe we can charge a little higher just because of what it can do more quickly, or more effectively, or more efficiently. But that's going to be a sales job that has to be done to the VC, and you're going to have to convince that wherever you're getting the money, whether it's a VC or one of the large manufacturers that are actually doing this, right? And certainly the Boston area has some fairly large manufacturers that could do that, right? So it's who do you go and convince? And I think having a plan and an ID that in fact is can be promoted would be really important, right? And it could be the VC, but it really could be Millipore for all I know, right? they have multiple new devices, and that in itself might actually be a reasonable place to approach. So it's a little balance. It's who you find interest.
Okay, awesome. So there are a ton of also I think pretty technical questions that we've gotten through the chat here. There's kind of a theme of several questions. Paul Banks, a couple other folks on sort of airborne virus. And for example, how are we able to monitor the air quality of indoor spaces that might contain the virus? Is there let me find this other question I saw down here a little bit further before. What are the, what understanding do we have of airborne transmission of the virus? How do your two technologies fit into that? And then maybe Anne-Marie, you could sort of weigh in on your perspective on airborne transmission from the healthcare perspective.
Okay, maybe I'll jump in first and just talk a little bit
fluid dynamics, man.
About the fluid dynamics. Sure. So so there is still some question about exactly how the virus is transmitted, and Anne-Marie will probably correct me when I'm wrong. Many viruses are transported by large droplets, which happen during coughs and sneezes, or even when you're vocalizing, and you can break up spit and other things on vocal cords and between your lips and so on. Those are typically not as transmissible as COVID-19, and I think a lot of people think that one of the reasons is because that COVID-19 is actually transmitted by much smaller droplet sizes. And one of the reasons that becomes important is because the small droplet sizes, the small drops settle much more slowly in air than the big drops. The big drops will come out of air very quickly, and so it becomes sort of more transmission through touch and infection on hands and fingers and surfaces, and less so much on respiratory and breathing. And so that is a big challenge. When you come to trying to capture it, for instance, that's one of the reasons our design is as it is that one of the challenges is to capture not just large droplets, but small droplets. And from a fluid mechanics standpoint, we had to design our system specifically to capture droplets that were even smaller than a micron, things which could be even single virus size, which can be down to about 50 nanometers. So actually really quite small. And that's a challenge to do. They tend to avoid filtration, right? Because they've moved through smaller pores. That's why you need to go to an N95 mask in order to actually stop all the virus from getting through, rather than just a typical surgical mask. So capturing these viruses actually turns out to be really hard, because you have to have them, whether it's on my device, where we have a cooled, specially modified surface that we condense liquid on, and then capture the virus onto the condensed liquid, and then move the liquid away and then collect the liquid afterwards. Or even Jessica's surface, where again, you're really looking for impaction of those viruses onto a filter, or onto a material where they can be captured. You really need that impaction, and that's hard to do with the really small particles. So that's actually a challenge. But whether it's Jessica's technology or mine, both of those technologies actually allow for sort of sentry systems, where you could put them, say, in a ventilation system, right, and put Jessica's filter across, and then pull the filter out on occasion. I'm talking for Jessica; she'll correct me again. And then test the filter. In our case, we have a sentry system that continuously condenses liquid onto a surface, and then that drains into a test file, which you can take out on the hour, every 2 hours, every day, or whatever frequency you want, and go and test that either with PCR or some other technology. So that's sort of phase 2 of our system, is actually going from individual people to actually sentry systems that you can test environment for the virus. So Jessica, or
Jessica, you want to weigh in?
Yeah, so I think it's easy to assume that particles get removed by like a sieving. So, you know, you can think of a strainer and the liquid goes through and the solid bigger things stay put. And that's just not how these kind of masks and filters are working. So what we're making, I kind of, well, very, very briefly described, it's like a very, very soft Kim wipe. The reason why it feels so soft is because it's made of these individual fiber strands that are 1/100th of your hair. So there's thousands of them everywhere, and they have a very puzzle-like path. It's very tortuous. So stuff is like, the particles are bounced around so vigorously. And actually, we are insanely good at removing very nanometer sized particles, 50 nanometers, something like this. We cannot remove micron size. They are, maybe they're too heavy, I don't know, they go through. And that's actually, you know, not our goal species. So yeah, the technology is quite different. I will echo that using a mask not designed to pick up these particles is better than no mask. I definitely believe everything helps. And then I guess just to also add in, so we have this very, I can't say too much, but by having this kind of wet surface or this immobilized liquid layer on top, it helps to trap it in further. And this is a limitation, as was noted in some of the comments. There are many limitations to N95 masks because they were not invented for this purpose. They are, you know, an older technology that never needed to be reinvented, and they work great until a new problem comes up, right? So we are working on seeing if this special liquid that we're using, can it be applied to those masks? Can it be applied to, say, a coffee filter that many of us have at home, and how we can actually use this, the liquid layer in our case is more important for trapping the virus particles. And then we could just hand that over and say, analyze this. And they're very easy to kind of like oil and water don't mix. You can take this oil and it will easily separate off of that filter material. And the filter material could be used again with this new oil. So yeah, maybe Anne wants to correct us on.
Yeah, maybe Anne, you just sort of weigh in on the airborne threat and, you know, where you're seeing things from where you sit.
Well, I think you know, I see it as an airborne disease more than that you're going to get it from countertops or groceries or whatever. But clearly, it's a different particle size, as you mentioned, Jonathan, than it has been for other viruses. And you know, it is now being known to stay in the air, and so that air exchange is really important. And whether it's the air exchanging out of the room or you exchanging out of the room, that's the key for a lot of places and a lot of contagion. I think you know, one reason actually I think that nurses got much sicker. I wouldn't say more nurses got sicker than doctors because I really don't have that data, but I will say that at least in the ER, doctors didn't have to stay in the room as long with patient, and clearly infection is related to exposure, time exposure, as well as patient exposure. So there are a lot of things going on in the dynamic of the environment that you're in when you're exposed, but and also clearly household contacts are really mostly where people are getting it. So singing and vocalizing and moving your breath clearly has been shown to be the risk. I would be concerned if I was having to work in a big building that I didn't know what the air exchange is now. And that's places where, you know, I think of filtering systems and what are the new air exchanges and what's the HVAC, whether you're sitting in an operating room or a lawyer's office, wherever you're working, it's your time of exposure as well. But all of these, certainly N95s have been around for a long time. Certainly hospitals were mostly designed around isolating TB patients. So every ER has one or two reverse ventilation rooms but it wasn't prepared to have a reverse ventilation room for 7 or 8 patients out of 10. And in that, we saw some innovation using UV light to clean, using air filters right placed in the patient's room for every patient we went in to see, for closing down rooms and limiting exchange and limiting visitors and limiting exposures, doing more systematic changes to battle the problem. But I think there's a lot of great ideas here, you know, and 95 is what we had to work with.
Right. I'm kind of curious. Oh, Jessica, you're gonna.
Yeah, I just wanted to add in. So part of the question was what was happening in the UMass building. So before we were able to reopen, there was a huge spreadsheet about all the air handling capabilities in all of our labs. And that's actually how our reopening, total number of people allowable in the space in general. And then in the time of COVID, how many people were allowed. So I really love this comment from KP about, people that know about working in clean rooms with air handling, they need to step in. That's precisely what we're doing. So, I mean, we're also working with environmental health and safety and the Department of Environmental Health and how air handling is being handled in general. I mean, of course, the issue is that it's expensive. We've just seen this outbreak, in Canada from this spin class workout group, and best case recommendation is, okay, basically you need laminar flow between each spin bike. Not sure that's going to happen, but you're totally right. And at least at UMass and our fantastic students working in the labs, they well know that the safest place is to be by a chemical hood, by a biosafety hood, because that's going to pull the air away from them. Too bad we can't put them everywhere. So
Yeah. Thank you, Jessica. So Bob, I'm curious, this kind of raised a question in my mind about the sort of the investment ecosystem. Where do these kinds of the intersection of let's say biomedical technologies and building technologies, is there, where would that happen? Like where if Jessica or Jonathan would come up with an idea, would be something actually from a building technology point of view, even though it might be based on biomaterials or some other innovation that is happening in a different domain.
So a building technology focuses on organisms that are essentially 0.2 micron in diameter. What we're worried most in the building is, can you maintain a non-sterile product completely sterile from microbes, right? And so there's a big difference between the 0.2 micron and the 20 nanometer or 100 nanometer, virus. And that really, when I was working actually with making AAV, right? So that's a 25 nanometer issue, and dealing with getting rid of that or making sure it's pure was a different problem than how you actually dealt with what we were making as protein that's microbial free. So most of the buildings are really focusing on microbial issues, right? Usually not necessarily delivering air that's microbial free, but relatively pure without particles which sort of have things gone there. And then certainly liquid is the 0.2 micron. But the virus is different, right? And given that they can be much more hydrophilic, then you can design something to potentially screen them out just based on that. And so it's physically a different problem compared to the room itself.
Right. I'm curious about the investment ecosystem for that, though. Like, is it an entirely different community, or like, are those communities talking? Like if you have an idea that's really coming out of the biotech sphere, but would be for a product that's going to go into, building technology or HVAC systems or something. I'm just curious how those, investment ecosystems interact with each other, or if they do.
So I'm not aware of a whole lot of interaction. The HVAC system folks, basically you go to technology, companies with that technology, and they design it for a facility you're actually making a drug or a sterile drug. Like I said, the folks that probably have the best sense of that are the folks that make filters, right? Whether it's Millipore or the other ones that are there, Sartorius. Those folks probably have a better sense of the interaction of the material with small molecule, and they are making viral removal, and whether they're multiple different ways you can do it, but it's more a question, well, how are you going to remove that, and how the viruses interact so that you can get rid of them, right? And the filter manufacturers have the better sense of that. It's just not the HEPA filters that you use to actually make it relatively bioburden free, just engineering wise. There's somebody there that actually knows the details, but most of it are just installing the equipment, because that is the most expensive part of actually making a room that can make a protein bioproduct.
Yeah. And then, of course, is the cost equation, as you said. I mean, can these
Absolutely.
things be done in a way that, it can be scaled to, a clean, semiconductor clean room, or, a NICU that has to be very, very pure. Those things are very specialized, right? But you can't really scale that up to, a large campus or a large office building or something like that. So no, it's really, really interesting. There's I guess I wanted to, there's a ton of other questions in here as well, but I wanted to maybe talk a little bit about engineering education and how how the fundamentals we teach in engineering are part of this innovation process. How did some basic fluid dynamics or basic biomaterials ideas that you might teach at the how did the education process fit into what's happened here?
For me, actually, the education process, and I had a slightly different education background, but it's really for me, what was most beneficial is sort of taking a problem and breaking down to the component parts. And that's often what is done within engineering, regardless of what discipline, is breaking a problem down to something that can be tractable and measurable, right? And that's often the difficulty for people that are not doing that, whether it's biotech or some other aspect of engineering within the natural world. And it's the ability to do that, whether it's a problem set or a specific problem you're dealing with that you don't have all the answers. How do you lay that out so you can accomplish at least steps toward the goal? And if it doesn't work, you have alternatives. Right? And I think that's an aspect of engineering that you learn just by dealing, whether it's problem sets or dealing with problems that are presented on an ongoing basis. And using that generally is really helpful and not always used by everyone. And so to me, that is actually really helpful as far as the engineering discipline, if nothing else.
Absolutely. Jonathan, Jessica, how does
if it's helped, sure. Absolutely.
Talk about this from the classroom point of view. How can how does both your educational process or your teaching methodologies help the next generation do this kind of thing, or vice versa? How does the education or the fundamentals of the classroom lead you to these kinds of innovations?
Yeah, you want to go first, Jessica, or do you want me to?
Yeah.
Sure, I'll go ahead. So, okay, last spring, if we can remember that far back, I was teaching a biomaterials class. I had a range of students from many different undergrad engineering departments, and interestingly, students always say that they don't hear enough about current events. And of course, we love to integrate current events and lifelong learning, things of this nature. And so the class before spring break, we had articles from the New York Times and CNN all about this crazy virus coming out and what the best case recommendations were. Biomaterials class, so we read about masks. Do you need masks? Do you not need masks? Virus development. And we had this really awesome discussion. I would honestly say those fantastic seniors in chemical engineering knew more about virus manufacturing than I did. And they were like, this is crazy. We're not going to have a virus in a week, or a vaccine in a week. Sorry, we certainly have a virus in a week. We're not going to have a vaccine in a week. And they're like, oh, well, how is it developed? And then they would just teach each other about that. And I would say, okay, so next week you're going home. It's going to be spring break. How about you start to talk to your families about how to interpret these kind of news articles? Many of you are the first generation engineer in your class. Like, even if you don't become an engineer, which of course many of our students do not, it's still kind of our job, as was said, to help other people understand science, right? This is what we want to kind of take home. I can still read through all these news articles and say, okay, maybe this one is true, maybe this isn't so true, and things like that. And they all thought I was crazy for even reading these articles. And then no one came back after spring break. Campus shut down. And that first virtual class, I would say 90% of the students had the little coronavirus emoji as their backdrop, fake background kind of thing. And they said, okay, now we're paying attention and trying to figure out what we can do to help. So in some ways, the education is both maybe Jonathan will have some fundamentals that he wants to integrate in, but it's also like just being aware of what's going on in the world around you and just drilling in the importance of, respecting society. Because of course, at first we were saying, okay, younger people, student population, they're not really getting sick, but they're carriers, and yeah, just general awareness.
Are we in the, I guess in the chemie curriculum and biomedical engineering curriculum specifically, are we teaching vaccine development in some way? Are the students getting some perspective on that? I know you're ramping up a biomanufacturing senior lab in chemie. Is that something that the students are getting somewhere in the curriculum? Yeah, or
I mean,
synthetic biology.
Even I'm on a few different projects where we're doing CRISPR and we're doing all these different kinds of vaccine development and things like this. But my undergraduate, engineering degree, we certainly did not talk about that. And I would also say, I'm teaching kinetics and reactor design. Historically, that was petroleum. Let's scale that up, Exxon, this and that. And now those aren't the questions that we're asking about, right? I mean, everything is manufactured at scale. So let's talk about all sorts of different, examples of what we're making and how much it's going to cost and things like that.
So I'll add a little bit of insight from my side to Jessica's discussion. So one of the things that I've noticed is that, sort of the fundamentals of a lot of what we teach and the way to understand, the viruses or even the technologies which we're developing, starts really young. And I'll give you this example. I do this outreach program where I go out to middle school and early high school kids. I've got a lot of different versions of it. And one of the versions that I have is all about the interaction between liquids and solids. And it's just about getting kids to create droplets and moving the droplets and looking at how they interact with different solids. And we do floating on liquids and droplets on different surfaces. And we talk about contact angles and, moving and hydrophobicity and all these sorts of things, which are really easy to see sort of graphically, or in your hand, you can get a feel for it, you can experiment with it. And we have a lot of fun, with like 10 different demos that I have them do. And what I was thinking was, those ideas that I'm teaching middle school kids are exactly what I'm actually implementing at a slightly higher level, like in our technology, right? With the physics and the science and the fluid dynamics and all the differential equations. But really what we're doing is we're trying to understand how do we capture the liquids, how to make them interact with the solids, how do we design a surface that best interacts with them. And that's, that, first of all, is really interesting and fun to me to realize that this knowledge starts really young, and the intuition of how these things will occur, or the designs that you can create, can be done at a really, at a, without having all the higher level complexity of, a PhD program. And so, when it came time to start doing some experiments in the lab, I had undergraduates. I had 2 undergrads who've been working with me since as senior thesis projects, who started on one project, and then we transitioned to COVID, because in part because their projects were similar enough that we could transition, and in part because we needed to do things remotely. And so I had ideas of how to do it. And once they heard I was doing something with COVID, all they wanted to do was work on COVID. And that's one of the really nice things about, working with something that's topical, not necessarily a good topic, but it's still topical, is that you can motivate people to do work and give them a project that actually gets them excited, because they feel like they're making a contribution and a difference. And the difference level in terms of enthusiasm, I could give 2 students the same exact project. One is motivated by COVID and the other is motivated by nothing but basic science. And you'll get twice as much done with the proper motivation. And I think that's really something you learn and you take back into the classroom with you, is that teaching science as equations and not teaching it in terms of application. Where is it useful? How can what I'm learning right now, make a difference in the world? That's the way to teach and get kids interested and excited. And it works both in the classroom and in the lab.
Yeah, the Apollo program, right? This is kind of our Apollo program in a way, maybe, way more expensive. I think you're exactly right. And in fact, one of our, one of the other challenges we have in STEM and in engineering is the lack of women, lack of underrepresented minorities that go into this field. And again, not that it is a good thing, the virus itself, but the ability to make an impact on society, to solve a problem that's going to make a difference to their communities and to their families, I think can inspire kids to go into engineering. I think medicine has beat us there, at least as far as women are I think it's more than 50% women in medicine, but engineering, we've got more work to do on that front. But Anne-Marie, you want to weigh in on engineering education and how it can how it led you to your place, but also maybe how it can help us solve these problems in the future?
Well, it's been a while in engineering for me, but one thing I do recall as a senior, sort of at the end of our year, they pretty much said to us, I remember, I think it was Kittredge who said, okay, now we've taught you everything, now go out and do it. And we're like, wait a minute, we're not ready. And I think that's obviously before internships and all that stuff. But I think, need to know and wanting to contribute are great motivators. And I can tell you it works on me daily in the emergency department. That sort of need to know motivation is just keeps you sharp and keeps you going and keeps you interested. That sort of short-term outlook isn't everything, because you need to stay with the data, you need to do the research, you need to do the right thing, you need to verify and take time and, really not just be reactive. But I think it's a much more informed youth these days. I have 2 college kids actually, and, they're much more integrated, partly through technology that we didn't have as kids. And, I think there's a lot of opportunity for kids and a lot of opportunity for women. I think, the doors are falling down to open to women and minorities. So I think it's just a matter of time, but it will take effort too.
Yeah, it does. And I think that's one of we have all these technological challenges and these health and these, grand challenges that matter to the planet, the virus, not the least among them, but climate change and sustainable energy and the clean water. There's all these things that engineers can impact. But we're leaving a lot of talent on the table, and so we've got to, I think we've got to do better. And I think medicine has, to a certain extent, shown us the way.
But we
One
also,
point-
what you have now is
Yeah.
what we didn't have. You have models. There was no Desico when I was in chemical engineering. There were great professors, but there are models and models throughout the world too. So I think, and when I went into medicine, there weren't as many models either, and now there's a whole generation of models.
Right. Yeah.
We're making progress.
Yeah. No, thanks. Oh, Bob, you were gonna
I'm
try.
just saying,
Yeah.
right now, co-ops and interns, often within the chemical engineering discipline, is in 50% of the folks we have within Cambridge. And it's just because I think the cross between the engineering and the biology has been very attractive for a much broader audience comparatively. And that actually, it's been interesting to see that many chemical engineers that certainly have gone within Cambridge to certainly the co-op programs that I've done and the intern programs we've done.
No, that's a great observation. In fact, KP was mentioning in the chat here about the importance of those experiential learning opportunities for students to actually go into companies or into research labs and really, we can say all the things we want in lecture, but that hands-on, that experiential experience, whether it's in a research lab on campus or in a company, I think are super important. Let me ask you another diversity question about the entrepreneurial world. Are you seeing the same level of women and underrepresented minorities becoming founders of companies, or is there still? I know we hear from Silicon Valley that the founders are overwhelmingly white or Asian men, but are we starting to see, at least in the Boston, Cambridge sort of ecosystem, more women and minorities starting companies?
What I've seen is more. It's still not to the same extent as essentially the engineers got to come through, but there's certainly more female CEOs within the biotech than there used to be, and that's just based on what I've seen and who I know. Some of them are actually people I worked with that have gone and are the head of multiple times, right? And these are folks that I'd worked with when I was at Pfizer. So I've seen it. They just don't represent the largest majority. And curious enough, the VC groups are encouraging that as far as how they're doing it. And that's actually been helpful because you need that VC money to sort of start a company, whether it's a device or a new drug. And that's important.
Well, thanks for that perspective. Unfortunately, I know we didn't get to a number of the questions that were in the chat. I really appreciate everyone who joined us in this conversation. I really specifically, want to thank Jonathan and Jessica, Bob, Anne-Marie, for taking a really, a significant chunk of their day and over a couple of days to get ready for this event and to share their experiences with you all. So, thank you so much for participating in this. Thank you to MK and Karen for your support as well, and Alex as well for taking time out of her busy schedule as a biomedical engineering student to help support this. This has been a really, really fun experience. I think this is the tip of the iceberg of the kind of thing that we should be doing more with our alums and with our extended UMass family. I think we're starting to think about education as, and I think Jessica mentioned this earlier on, a lifelong learning process. You don't just stop learning when you walk across and you get that medal and you get your diploma and you move forward. You keep learning throughout your life. And so UMass should be a place where you can keep coming back to for things like this, for discussions like this, for interactions with fantastic professors and alums and our industry partners. So, I think this is an example of the kind of thing that we can do together as a UMass community. Look forward, certainly from engineering to do more of these and create more opportunities for our alums to engage and to participate. I'll just, as I'm going off the stage here, mention our outstanding alumni awards from the previous year. These were to have been awarded in the spring of 2020. We had a big event plan. They were all going to go to the senior design competitions in MIE and ECE, and they were going to visit with students. And of course, COVID came, and we weren't able to do that. We're keeping our fingers crossed that we might be able to honor these folks, and as well the next class of outstanding alumni that we'll be selecting over the coming months. And hopefully have an opportunity to recognize them this coming spring in 2021, knock on wood. But if not, we will do it virtually, and we will carry on. I want to again thank everyone for participating. Keep in touch, keep your ideas flowing to us. We look forward to talking and answering your questions. Any questions that we didn't get to, please, shoot those to, certainly you can shoot them to my email here or to MK, and we will try to get back to you or get the right people in touch with you. Keep coming back, continue the conversation, and thanks everyone. Looking forward to solving the pandemic and getting to the other side of this and being a healthier and safer community. And just thank you so much for everything you're doing. So goodbye, folks. Thanks again.